Okay, we are ready for the YouTube live streaming. So whenever you're ready, you can just start. But it's good to start with the recitation of Kitab Al-Fatiha. Okay, off you go.
Hello. Okay. Assalamualaikum. And today we will present about PBL 1 Trigger 2. Trigger 2 is after 1 week, Gopal started to develop fever, headache, and itchiness sensation at the bite side. He also experienced seizures, and upon laboratory investigation, he was positive for viral infection. Okay, for the FILA table, F. Facts. Develop fever, headache, and itchiness sensation at the bite side. Experience seizures, positive for viral infection. And for the ideas, we think that Gopal might have rabies. Learning issue, fight and flight response, common symptoms of rabies, mechanism of blood clotting, pathophysiology of fever, difference between viral and bacterial infection, diagnostic test for rabies, the treatment of and management of rabies, and Islamic perspective. And for the action plan is to explain the fight and flight response, to identify the common symptoms of rabies, to discuss the mechanism of blood clotting, to learn about the pathophysiology of fever, to investigate the difference between viral and bacteria infection, to study diagnostic tests for rabies, to learn the treatment and management of rabies, and to relate to Islamic perspective.
If you got only one idea, what do you mean by ideas? Why is it only rabies?
Because based on the trigger 2, we managed to conclude that the most possible case is rabies.
Not bacteria.
No, because positive for viral infection.
So positive viral for viral infection is not positive for bacteria. Anyone? Think.
Yes, they can get bacteria infection too.
You think it's possible to get the bacteria infection?
Yes, like throat body strep infection, which is a bacterial infection, which you can feel throat is sore and scratchy.
Nasratul Hamizan, you agree with Fakihah? She said that it is also possible to get the bacteria infection. Do you agree with that?
For me, I think no, because if Gopal was infected with bacteria, we will call it as bacterial infection.
Okay, so is it possible to have like both at the same time? Camelia,
No.
Camelia. Oh, Nasratu said no. Camelia, do you agree with Nasratu? She said no. My question is it possible to have both infection at the same time, or it's not possible?
It's possible.
Why do you think it's possible?
Because bacteria and virus have everywhere.
You can find everywhere, right?
Yeah.
And can the same person have both infection at the same time?
Yes.
Is it possible?
Possible.
Okay. Ashikin, do you agree with Camelia?
I don't think, doctor, because
Why? Yes, why?
Because I think for the viral infection, it's caused by virus.
Only by
I don't
virus?
Yes.
But the question is it possible to have both infection at the same time?
No.
It's not possible.
Yes.
Okay. Huda, do you agree with Shikin?
For me, I think maybe Gopal can get both infection because as we know, in dog mouth, there are a lot of microorganisms which is virus and bacteria. So I think maybe Gopal can get both infection.
You think both, yeah? And Alia, do you agree with Huda?
Yes, I agree with her.
Why do you agree with her?
Because
Because of what? Because he is your friend. No, she is your friend. Atika, what do you say? Is it possible to have both infection at the same time?
I think it's maybe possible, doctor.
Maybe possible. Why do you think it's possible?
Because when it diagnosis, it maybe have the bacterial infection too, but it cannot be analysis at the hospital.
What do you mean by analysis?
I mean like macam masa diorang diagnose apa infection yang Gopal
English,
tetap
Atika.
when they diagnose what infection that Gopal have, I think Gopal maybe have bacterial or viral.
How do
But
they know?
when they have diagnosed that Gopal have viral infection, but it has possibility that Gopal also have the bacterial infection too.
Okay, if you look at your third box in learning issues, point number 1, 2, 3, 4, 5. You mentioned that there's a not different, okay? It's difference, CE, not T. Difference between viral and bacterial infection. So how we can differentiate between these two? Based on what? What is the criteria that we look for if you want to differentiate between the bacteria and viral infection? You as a professional nurse, when the patient come to you, co-participants come to see you, and how do you evaluate him? How you look at him and know this is a viral or this is bacteria? Based on what? What is the fact that you can know to differentiate that this is viral and this is bacteria? The answer is in the screen. Come on, everyone. Nabila?
yes, I think it's because the symptom is different.
Exactly. So brilliant. We look at the symptoms. So if we look at the symptoms, we can narrow down our diagnosis. We can know because each viral and bacteria infection, they have a different sign and also symptoms. So the first time patient come to you, you already know that what is, you have like a prediction, what is going to happen to him. What is the diagnosis? Or in other words, we call it as a pre-diagnosis stage, where you can predict early. Based on these signs symptoms, you look at it, okay, maybe fever is quite common. Fever, everyone with fever, like flu virus, coronavirus, fever, headache, itchiness. But for some people with diarrhea, for example, now you know, oh, maybe food poisoning, right? So this is your stage, early stage of diagnosis. Oh, maybe something to do with the food. Maybe the food is contaminated with the E. coli or bacteria. Okay, but the distinguished features for you to know, to classify that this is the viral infection is what? What is the unique thing in the sign and symptoms that you can find that makes it stand out? It's point number 2. Experience seizures. Seizures is like different from the other bacteria. Some bacteria have this one, but it's, you know, it's narrowed down your investigation. Because if you got bacteria, the most common salmonella, for example, E. coli, based on your food that you take, or poisonous food, expired food. But diarrhea is not there, but it's more towards the CNS. The seizures is affect the central nervous system, and you're gonna, you know, paralyze for a while. Okay, so this is one of the things that you need to investigate further. And this is the distinguished features. This is very unique. All right. So when I ask you how to differentiate for the learning issues, for number 4, difference between viral and bacteria, you look for the different symptoms. Okay. All right. We'll go to more detail, I think, after this. But I think it's also good if you can include why you put in the ideas. Maybe you want to add the bacteria too, because these stray dog, maybe they are not non-hygienic. Okay, so they are not exclusively affected by the virus. At the same time, it's also possible to get infected by the bacteria. Okay, so usually it comes with the bacteria because this is the most common pathogens that can reduce or that can compromise the immune system. So their body protection is low. So after that, it's easier for the virus to come and invade the body system. Okay, so this is good. So we move on to the next slide. Fakiha, what is the fight and flight response? Yes, Fakiha, unmute.
Yes, sir.
What is fight and flight response?
The response of the body to defend ourselves from after getting the symptoms.
So what is fight? What is flight?
Fight is how our body respond and counter back the bacteria, and fly is how our body, what you say, how our body
Fly.
no, how our body responds and stay and keep getting better.
Khalila, you agree with Fakiha? She said fight is counter back, flight is getting better.
For me, I think fight is like the body like help the system like help the body to be able like strong enough to like fight back maybe. For flight is like to get away. I think. Because in the like in the situation like Gopal is like being chased by a dog, right? So flight maybe is to help Gopal to run away. The flight, I mean. I think. Is it?
Atika, do you agree with Khalila? Do you think what's your thought about fight and flight? What is this? Why the name, why the response is so catchy? What does it mean? I didn't see that Gopal fighting with a stray dog.
Maybe the fight term is referred to how Gopal, how Gop how the Gopal impulse send the sign to Gopal to run away from the dog.
That is fighting. That is fight.
Maybe no, sir.
Okay, Alia, what do you think?
I think a fight and flight response is physiological response. When we have strong fear, strong emotion like fear.
Does it comes together at all times, or we can choose fight only or flight only?
Together, they come
They always come
together.
together.
I think
Nasratul, you want to fight or you want to fly? You want to fly? Which
I
one?
think both. For fight, maybe after has been diagnosed with viral infection, we how our body responds to how I will respond to the infection. And for flight, maybe when the Gopal was chased by the dog, he
And
is
Gopal fly.
running away from the dog.
Okay. Okay, we'll see what does it mean. Camelia, do you have any other thoughts? What is fight and flight response?
I thought it seemed like that. Fight and flight response is when we respond when we have got something. So
Okay, give me example. Give me example where we can have the fight and flight response apart from Gopal.
touch hot kettle.
Again, Camelia,
Touch hot kettle.
what is it? Shout, Camelia. I cannot hear you very well. You say
Touch string iron.
Iron. When you ironing your outwear, everything, yeah. So what happened?
Tarik tangan cepat-cepat.
Oh, when you
Bila
got
panas.
when you touch the heat plate, yeah. Okay, so you will have like a response, like a reflex, and you stay away from that. Give me other examples of the fight and flight response. Anyone?
Getting pricked by a needle.
Again, Fakiha?
Getting pricked by a needle.
Needle prick. Okay, correct. What else? What else? Give example. This is most common and it happens almost all the time in our life.
When we almost fall,
Okay, what happened?
we will try to grab anything at our
Right.
surrounding.
You will
Try
cry.
to grab anything at our surrounding to stabilize ourselves.
Okay, when you're about to sleep on the floor and then suddenly you grabbing on something. Okay. Okay, what else? Shikin? Give example, Shikin.
Yes, Doctor.
Give
Ni,
example.
when we suddenly stop, kejap.
Stop from what?
When dekat, macam kereta tu tiba-tiba datang dekat kita, kita terus stop.
Kereta apa ni?
Bukan, contoh macam kita melintas jalan ke apa kan, lepas tu kereta tu laju, tiba-tiba datang kat kita, lepas tu kita pun macam, tiba-tiba macam Macam mana? Macam reflex ke belakang ke atau mengundur ke berlari ke macam tu.
Okay, I got
Ah.
it. So you're afraid that the car might hit you,
Yes.
so you stop.
No, I run,
Stop,
I
then
run.
the car will hit you.
I run.
So not stop. If you're in danger, you have to flight. You have to fly.
Yes.
But sometimes at this time point, you cannot decide because it occur spontaneously, right? So you don't have this, your brain is not prepared for the emergency cases. So it's like a response, it's an immediate responses, and you are maybe the information is a bit delayed to be translated to your brain, right? So that's why you think that, oh, you in Indonesia we call it kaget. Is it kaget? Right? So you stop for a while, and then you don't know what to do. Okay, so sometimes it occur. That's why it's quite dangerous. If someone have a lesser amount of responses, okay, or reflexes, they are not the same between people. Like Camelia, maybe she's a bit sensitive with the iron plate. Oh, sikit dah. Okay, but someone like maybe Alia, she can hold it longer. Is it true, Alia? No, just joking. Okay, next slide, please. That's good. So this is one of the examples. Okay, it's kind of a safety mechanism for your body to protect your body system. Okay, if you are going to a climbing, like mountain climbing, right? So you go to the higher level of the mountain where the oxygen is a bit saturated. So you are gasping for air, right? So what happened is that one of our cells or alveolus or bronchus, they start to expand a bit more to allow more oxygen. Okay, so the diffusion of the oxygen might become slower, because if it happens like now in Earth, okay, on the ground, where you can get the oxygen readily. So in the mountain, the oxygen is lesser, so the body have their own mechanism to protect you from dying. So your rate of breathing also being controlled. It's not as quickly or as we are in the ground. Okay, so this is one of the things that is to protect ourselves. It's a safety mechanism. Like Gopal, if Gopal stay there, okay, maybe the wild dog already eaten him, for example. Okay, so sometimes this is not being you will not have the courage to decide what you're gonna do. Sometimes it just so happen. That's why I say Gopal is quite very, very good in this case, because he can manage to run away. Yeah, so it's safety mechanism for him to keep him alive. All right, so in everyday life it always happen in ourselves, but without us knowing that. So it's a reflex mechanism. In terms of the infection, for example, your body is struggling to get rid all of this pathogenic infection without you knowing, or without you telling your cells, telling your body to do that job. Okay, so it occurs naturally. And you see here, sensory information is gathered by the eyes and converted to electrical pulses through specialized cells, which are known as photoreceptors. So this is you trying to say that first time we look at the possible risk or the danger, and it perceive, or they send or relay that information into the brain to be translated. So it must have the sensory receptors or photoreceptors to capture what is the risk or what is the danger. Okay. Next slide. Okay, the second part is the brain. Who's gonna tell me about this?
Yes, sir.
Yes, go on.
When the information converted to the electrical impulse to the specialist cell in the eye, which is photoreceptor cell, the electrical signal is then transported by neuron to the brain. To brain processing, the second step. Information from the sensory organ travel to the thalamus in the brain. The thalamus is considered the relay station of the brain, collecting information and sending it for processing. The thalamus will send the information to one part of the brain called amygdala. Amygdala is responsible for processing emotion and fear. The amygdala will assess the situation and will then send the information to the part of the brain called hypothalamus. The hypothalamus controls the sympathetic nervous system and the endocrine system. This result in the hypothalamus release corticotropin releasing hormone, or we can call CRF, to signal the pituitary gland, which is an endocrine gland, to release adrenocorticotropic hormone, or ACTH, into the blood. The hypothalamus also activate the sympathetic nervous system. Yeah.
What do you mean by sympathetic nervous system? What does it mean? What is sympathetic nervous system? How do we classify? Yes.
The connection of nerves that help your body to fight and flight.
The sympathetic, and the other one is
Connection of nervous system.
there are two types. We call it parasympathetic and sympathetic. Yes. Okay. What's the difference between para and sympathetic? Why the process here choose to be regulated, or it's regulated by sympathetic, not the parasympathetic nervous system?
Parasympathetic happen after sympathetic nervous system, because parasympathetic helps our body to relax, to cool down from the sympathetic nervous system.
Okay, very good. So generally, I can say generally, sympathetic is the main activation. Okay, is a activation site. The functions is to activate, like what? Increasing heartbeat or heart rate, for example. Okay, increasing breath. Okay, not heart rate. Breathing pattern. Increase sweating. Okay, so parasympathetic works the other way around, where they inhibit this activity. They're relaxing or slowing down this mechanism. So there, when we talk about the nervous system, you must remember these two main category or two main type of a nervous system. Okay, so the sympathetic is the main one that is affected, that controls what the body responds when they have this information translated by the brain. Okay, so the brain, as I mentioned in my class, if you remember in the hormone class, yes, so we have the hypothalamus that produce this hormone. We call it corticotropin releasing hormone, and from this hormones, okay, they will travel down to the organs, okay, or they activate another gland or endocrine gland. In this case, it's pituitary gland. So this is the anterior or posterior pituitary gland. ACTH Secreted by
Interior.
anterior. Anterior, very good. So anterior, anterior pituitary gland produce the adrenocorticotropin hormone into the blood. Okay, now we will see what is the function of this ACTH. Next.
Okay, this is the third step of fight and flight response. We'll tell about how the message we send to the body. The anterior pituitary gland release ACTH, which travel through the blood and stimulate the release of stress hormone cortisol from the adrenal cortex. At the same time, the sympathetic nervous system synapses with the adrenal medulla and lead to the release of adrenaline or adrenaline.
Okay, so the ACTH is acting on the
Adrenal cortex.
okay, they will travel in the blood vessel and they will go to the adrenal cortex. So if you look at the name adreno cortico means adrenal gland at the level or the region is cortex, not the medulla. Okay, so from the name itself or the name of the hormone itself indicates where they are acting on and what is the location. So adrenal gland is an endocrine gland situated on the or located on the kidney, both right and left kidneys. It's a pyramid shape and the functions is to produce cortisol. So this is the final factors or final molecules that has been secreted in order to activate the stress pathway. Okay, so this is one of the most important things. Some people say that cortisol is as a stress hormone. When you are under stress, cortisol will be released into your bloodstream. Like now, some of you feeling stressed because I'm keep on asking you questions and you keep on looking the answer on the Google, right? So I understand that, but it's good for you to know that at this point, your cortisol level are higher than the normal state. Okay, once we finish this session, then it goes back to normal. So the sympathetic nervous system leads to the release of adrenaline from the adrenal medulla too. Okay, so can someone explain to me the adrenaline activity?
Okay. Adrenaline will stimulate the liver to convert glucogen to glucose so that more energy is available to the skeletal muscle. Adrenaline also stimulate the heart to pump at a higher rate to deliver a higher cardiac output to the skeletal muscle, and that is so they can nourish the glucose, oxygen, and nutrient to be able to work efficiently in the preparation for the response. It also stimulates the intercostal muscle to function at a higher rate to increase the breathing rate, which is then increase blood oxygen levels while removing carbon dioxide from the blood at the equal higher rate. Adrenaline also leads to the vasodilation of blood vessels to the skeletal muscle and vasoconstriction of the blood vessels to the digestive system so that more energy is directed toward the fight and flight response.
Okay, so we have discussed the effect of epinephrine in the previous session, right?
Yes.
Okay, can someone recapitulate what is the function of epinephrine? What is the effect of epinephrine on the on Gopal? What does it do? When it's been secreted into the system, and in this case, you say that it's a short-term stress. So what really happened to the body system? Look at the physiology of the body. What really happens? Camelia mentioned some of the information just now. Can someone else tell me? What does the epinephrine do to Gopal's body system when he's under stress? Yes, that's right.
Increase heart rate and increase breathing rate.
Excellent. Okay, heart rate increase, breathing rate also increase. Now it's a vasoconstriction, and it leads to what? Camelia, just now you mentioned about the vasoconstriction. It means vessel constriction. The blood vessels become narrow. The diameter becomes smaller. It leads to what, Camelia?
Higher pressure.
Higher pressure, right? Okay, so when it's higher pressure, what will happen to the breathing? The heart rate become increased. Now, if you have the water, the pipe, right? If you pinch it, so it will become pressurized, right? It's become pressurized and the pressure is very high now. Same goes to your blood vessels. With this type of hormone, epinephrine, it's acts on the adrenal gland, okay? It's been released into the body system, whether it's through the bloodstream or directly on the organs. It will constrict the blood vessels. And when they constrict the blood vessel, the flow of the blood will become high or low? Become okay. So the rate of the flow or the rate of blood, the speed of the blood that goes to your cardiovascular system is increasing. All right. So this is how the body responds. So you have more exchange or more pumping of the oxygen or the blood throughout the body. Okay. So if it's vasodilated, less pressure, so you become more relaxed. And both hormones works, as I mentioned in my class previously, it's a feedback mechanism. If there's no more wild stray dog that come and running or chase after him, so he can relax, right? So for the mechanism of relaxation, we call it as the feedback mechanism. Means it counter again. It goes back to the normal. So that's why if you look at the hormone, there are always hormones that activate certain pathways, and the other hormones try to bring back to normal, okay, or inhibit the activity. Like people with the hyperthyroidism, where they have an excessive amount of thyroid hormones. So there's always a feedback mechanism in order to maintain the balance of this hormone level. Same goes with the people with hyperglycemic or diabetes. You, at some point when you have your dinner, okay, your blood glucose level will rise, right? And your pancreas start to produce hormone, we call it as insulin. So what insulin did was they take up this glucose molecule back to the cells. Okay, no, you cannot go to the blood. Please come here. I want you to be stored in the cells. That's the job of insulin. Don't roaming around inside the blood system. Because if you go to the blood system, if it's high sugar, you will have the symptoms. Right? That's the diabetic. You check 7, 8, 9 diabetes. Okay, so in order for the body to protect this from hyperglycemic, right? So we have another hormone which is glucagon. So this glucagon will, what the glucagon did? Again? Yes, Kalila. Alia, anyone want to answer?
Glucagon control glucose level.
How does it control? Insulin also control glucose level, but how does it control?
Glucagon increase glucose level.
Okay, glucagon increase glucose level.
But
Insulin will
Decrease.
okay,
Glucose
it will decrease. Okay, so insulin will reduce your hyperglycemic condition. Your glucose level, your sugar level will be decreased by the activity of insulin. So the insulin, once you finish your meals, your blood sugar high, insulin will bring it down. Okay, but if it's too much secretion of insulin, the patient will have hypoglycemic, too low glucose, right? So in the case of hypoglycemic, the glucagon come into action where they raise up back the sugar level back to the normal. So there is always feedback mechanism. That is one hormone that, you know, induce or stimulate the activity. The other one functions to inhibit it. Right? So remember this in the examination. If I ask, so you have to illustrate the image or the figure how we can describe this activity. Okay, next. Okay, now you do the talking. I will just ask. Why stop? You don't want to continue. Whose turn it is? Is this? Why do you skip? You don't want it.
Already taught.
You already understand that. You think it's not needed, yeah? Okay, up to you. You choose which one is the best for you. Who gonna present this slide? Also, you don't like it. You don't know how to describe. Go back. Go back. So every single slide matters, okay? Go back. Don't skip. Why do you want to skip? Okay, tell me about this. It's okay, you can ignore the one that you have understand. Just focus on the one that on the left. Why do you put that figure?
Actually, sir, this is the final step of fight and flight response, which is the fight physiological reflex. Adrenaline will increase the glucose level, will increase heart rate, breathing rate.
What is gluconeogenesis? What is glycogenolysis?
The process.
Again?
Glycogenolysis is we break down the glycogen.
Okay, what is glycogen?
Glycogen is glucose that insulin
You said glycogen is glucose. What's the difference?
glycogen is a store form of glucose that made up of many benefit glucose molecules.
Okay, so it's a stored version of the glucose in the liver. Okay, so when you have much more glucose, so some of them will be stored in the liver, some of them will be stored in the skeletal muscle, some will be stored in the adipocyte or the fat tissue. So the one that you've mentioned here is the stored version of glucose in the liver, and we call it as glycogen. Agree? Okay, so now what happens? Why do we need to break it down?
To give energy.
Yes, to give energy. What else? You can break it down, for example, when you are fasting. So you depend on the stored glucose in order to activate the cellular activity so that you still can function even though you are not taking any food. So some of the stored glucose, they will be stored in the form of lipid too. Okay, in the adipocyte or in the muscle cells. So you need that for a source of energy when you are deprived of energy. And some of them is mentioned in the slide. You put it, maintain glucose in the bloodstream. So when you are hypoglycemic, when you have very low amount of sugar in the blood, so the liver will release this glucose, okay? They will induce the glycogenolysis so that the glucose level will be secreted, or the glucose will be secreted into the bloodstream and maintain the normal glucose level. And what do you mean by gluconeogenesis? Huda? Gluconeogenesis. Look at the name. What happens?
Storage of glycogen.
Again, what do you say?
Storage of glycogen.
Storage of glycogen. What do you mean by that? Why do we need to store the glycogen? It's a process, right? It's a process. So what is it, gluconeogenesis? From the name itself, you can know what is the process.
I
Process of
think
making glucose, breakdown product of lipid or protein.
Again, Alia?
Process of making glucose from its own breakdown products of lipid or protein.
Okay, so from the name itself, you already know it's the gluco, refer to the glucose. Neo means novel or new. Genesis is the formation. In other words, we call it formation of a new glucose molecules. Okay, same thing. This is coming from the breakdown product, same like the glycogenolysis, but the glycogenolysis is from the glycogen or the stored glycogen and then break down into the glucose. But the gluconeogenesis is the formation of new glucose molecules from their own self or from any other lipid or fat cells. Okay, so they are two different mechanisms, but both of them, the outcome is giving up to glucose. Okay, they will produce a new glucose molecule. All right, so that is how your body maintains a glucose level at a normal stage. All right, next. You have gone through the gluconeogenesis lecture before with Dr. Samsul, I think. Lipid or carbohydrate, lipid metabolism or carbohydrate. Yes, so you have to recall again. You look at that slide again. If you don't understand, ask your lecturer. Okay, you can ask during the class too if you're not sure. Okay, now what happened here? Where do you put this slide? You can put anything on the slide, I don't care. But then the most important is you know how to explain it. Make sure you understand it, even though you just put an eye there. Look, inside the eye, I can ask why there's a white spot and a black dot inside the eye. Can you see that? So even this eye picture, if you manage to relate with the condition that Gopal is having, that's more than enough. Okay, now explain to me, you want to relate the blood vessel and also the eye or the pupils. It's 9:55 now. Your brain, start to produce the melatonin and then you start to feel like sleepy and bored. You just want to go to the bed. Talking about the eyes, sleepy. Go on. Vasodilation. Anyone? Fagiha?
Yes, Doctor.
What do you want to show me in this slide?
For the eye, I was from the eye picture. When we are going through a shock or a scared situation, our pupil become dilated and which we cannot focus and our mind become blank because maybe because of our nervous system is getting triggered by the shock that we receive. So our eye will become enlarged. So it's basically one of the fight and flight response.
Very good. All right, so you see the normal and also the dilated blood vessel. But as we mentioned earlier, okay, for the fight and flight response, you mentioned about the vasoconstriction, but now it's the vasodilation. Where is the vasoconstriction? You mentioned about the increasing in the breathing pattern, okay, increasing the heart rate. And now it's the vasodilation. What really happened? Does the vasodilation and vasoconstriction occur at the same time or at a different point of time?
Different point of time because the vasodilation happened at parasympathetic after the sympathetic.
Okay. All right, so you say that the vasoconstriction occurs first and then followed by the vasodilation. Camelia, is that true?
For me, it occur at the same time, but at different part.
What do you mean by that?
Different, which is vasodilation of blood vessel at the skeletal muscle, while vasoconstriction at the digestive system. That is what I understand.
Okay, now there are two different things. Fakiha mentioned about the vasoconstriction that occur first, and after that, parasympathetic nervous system got triggered where they start to vasodilate. But Camelia tried to say that it occurred at the same time, vasoconstriction, but you know, this is at the different site. Vasoconstriction only at the muscle. No, vasoconstriction at the digestive. Vasoconstriction at the digestive system. Vasodilation at the muscle. Okay, so during the fight and flight response, what are the things that really happen? You know that the vasoconstriction causes the increased pressure in the blood vessels, right? And more blood will flow to the heart. And then the heart rate will increase. But why vasodilation occur at the muscle? Doesn't it compromise the speed of running for Gopal? Why the blood vessel dilated at the muscle cells, at the muscle tissue?
Increase transport of oxygen.
Again? Transport of what?
Oxygen.
What do you mean by transport of oxygen?
So the muscle can move more smoothly.
Who's that? Who's that? Say it again. Who's that? Oh yes, Fakia. Again, what do you say?
Because so the muscle can move more smoothly when Gopal is running.
Smoothly? What do you mean by smoothly? Because now he need more energy, right? He need a good amount of energy to speed up. So why the blood vessel need to be dilated at this time point? So of course, this is occur at the different site. Yes, both can occur at the same time, vasodilation and vasoconstriction. Okay, vasoconstriction can cause the breathing pattern to increase. Okay, they will shut down the digestive system. But at the muscle cell level or muscle tissue level, they will dilated. It will become dilated. The answer is there. Come on, everyone, on the slide.
Removal of cellular waste products. Breakdown.
Provide nutrition to the creations and glucose to the body to have more energy to run.
Again, Fakiha?
To provide nutrition and glucose for the body have energy to run.
Increase the transport of oxygen, nutrient, and glucose. Okay, correct. So that is why we need to dilate it. Okay, we need to dilate the blood vessels. Enlargement of the diameter of the blood vessel means you will allow more nutrients more oxygen, more glucose because of the increased surface area. Now the space is bigger, right? So of course you can influx more energy in the form of glucose. You can influx more oxygen. So the surface area now become bigger. So the body is preparing, okay, the muscle tissue is preparing to give the maximum energy for Gopal to speed up. Okay, so for the muscle tissue that is functioning for the physical running, so they have to become packed up with a good amount of oxygen and glucose. So that's why it needs to be dilated. If the blood vessel at this muscle tissue vasoconstricted, okay, if it's constricted means less oxygen, okay, less nutrient, less glucose. So Gopal could not run that fast. Okay, maybe he got like a muscle cramp just in the middle. All right, so that is the reason when you mention about the fight and flight response, there are some parts of the body functions to fight. For example, increasing breathing pattern, okay, fighting for that. But some other side is flight, means they are relaxing. Okay, the muscle layer or the muscle tissue, they are vasodilated. So both of these fight and flight response, they comes together at the same time. Okay, but of course this is at the different location. Got it? So that's why we cannot separate the fight and flight response. It must be together at all times. Okay, next slide. I don't think that's Gopal.
And this slide, I will tell you how rabies infect our body. Okay, we can imagine that this bird is a Gopal. Okay, Gopal has bitten by a dog and get infection bite wound. Specifically, the saliva of the infected animal that transmit the virus. As we know, in a dog mouth have a lot of microorganisms such as Lissa virus. The virus multiplies in the muscle cell, which is myocytes, for a week or months or even a year. It depends on the location of the wound, but there are typically no symptoms at all during this phase. After that, the virus bind to nicotinic acetylcholine receptor at neuromuscular junction. Nicotinic acetylcholine ni dia neurotransmitter binding into membrane elliptical depolarization.
What does it mean? It's quite complex. I cannot understand that. Tell me about neurotransmitter. What is neurotransmitter?
Neurotransmitter.
Yes. Not neutral. It's neuro, right?
Yes.
Okay.
Okay.
Tell me, what is it?
Neurotransmitter ni macam dia yang bawa virus tu pergi ke sel lain.
Okay, dia yang bawa. Atika, do you agree with Huda? Huda say it's like a bus or like a taxi to bring the virus in to travel. Do you agree with that, Atika?
Maybe I agree with Huda, sir.
Why do you agree with her? Because she is my friend, you say? No. Why do you agree with her? She said that neurotransmitter is like a carrier that brings the virus to travel or to be transported throughout the tissue. Khalilah, do Do you agree with Huda?
I think I agree.
It's okay if you hate her. Just be honest. If you don't like you say, okay, it's a different one. Maybe it's not a taxi. Maybe you just like bike or something else. But the point is, do you agree when she said that the neurotransmitter is like a carrier that brings the virus through the tissue?
I think I agree with her.
You agree? Or
Yeah,
you partially agree. It's not 100% agree. Nabila, what do you say?
I also agree with Huda, sir,
Why?
because our neurotransmitter is like the charge in our body, right? It's like a particle, so like it can carry the virus.
Now you
Yeah.
say it's a charge. Charge is not like a carrier now. It means
Oh.
that you don't agree. Alia, do you got any other definition of neurotransmitter?
Yes, doctor.
What is it?
Neurotransmitter are chemical messengers that our body can function without.
It's a Google translation, right? It's a
Yes.
chemical, a messenger, a very complex one, right? Okay. Fakiha, do you agree with that Google definition of neurotransmitter? Of course you will. But tell me, what does it really means? Okay. Ashikin, based on your understanding, I just want you to understand. It's okay, the reading you can do at later time, but I just want you to understand what is the what do we mean by chemical messenger? What does the role of neurotransmitter? Does it really carry the virus? Or the virus carry them? Or the virus carry the neurotransmitter? Which one? Or it doesn't have to do with the virus or neurotransmitter? My question is very simple. What is the role of neurotransmitter? Okay, what is it? Where can we find it? What is the function of it? Why do we mention them here in our case, with the Gopal case? Nasratul, don't look at me like that. Don't get angry. Okay, you can answer if you ready. What do you think?
For me, like when Gopal was bitten by a dog,
Okay.
the neurotransmitter will receive signal like in Gopal body. I have been bitten by dog, so it will send the signal to the brain so that the brain will produce response. And then Gopal will away, I mean like lepaskan kaki daripada gigitan anjing tu.
Okay, where can we find the neurotransmitter? Huda, again, because this is your slide. Go on, Huda. Unmute, please. Don't say something that is, secret.
Nerve cell.
Nerve cell. Okay, you can find it at the
Nerve cell.
What is nerve cell? Nerve cell. What is nerve cell here?
Mas.
What is nerve cell, everyone?
Neutron.
Sorry?
Neutron.
Camelia, what do you say?
Neutron.
Neutron. Neutron and proton and an electron. What do you say? Neurons. Neurons or neutron?
Neurons.
Are you sure? Yeah. Okay. It's a neuron, right? Okay, the function is to send the impulse or the information or the signals. So it has like your finger, your hands, it can attach or bind to any other tissue. Okay, it functions to send the information in a form of electric, yes, but there is a molecule there. We call it as acetylcholine. So that is the one that Alia mentioned, that is the chemical messenger. So throughout the body, when we mention about the central nervous system, it started from your brain. Okay, so go through your spinal cord. It branches out at each level of your body system. Okay, so apart from the blood vessel, you have these nerve cells. Okay, so the nerve cells is for the activity, the conduction, and how you want to grab things. Okay, how do you want to respond to the iron plate? So when you have the reflex, means you are having a good amount of acetylcholine. So you can imagine the neurons that travel onto the muscle tissue on your hand can sense the increased temperature of the iron plate, right? So the point that you touch the hot plate of the iron, okay, the acetylcholine will be released at the neuromuscular junction. Okay, that is the location where these chemical messengers started to transmit the information so that the muscle can react or give the response. Okay, the unique thing about this virus, they bind to the nicotinic acetylcholine receptors at the neuromuscular junction. So the virus, I think it's in the, what do you call it? The cyan color, like blue color, right? Like a rod-like shape. So it binds at the point where the nerve cells binds to the muscle tissue. So the virus take this information or take this opportunity, they bind to the acetylcholine, which is the chemical messenger, so that they can transfer or they can be transported throughout the body together with the acetylcholine. Got it? Yes, you mentioned it's like a carrier. Yes, it can cling on the nicotinic acetylcholine receptor at the neuromuscular junction, and after that they will travel into the central nervous system through the peripheral nerve cells. Okay, so they are very small, so they can invade or get into the nerve cells. They don't get into the blood vessels like the bacteria. Okay, they love to go because they are very small, so they know where they are going. They're not simply go randomly, because for this Lassa virus, they have a special receptors to detect and recognize the acetylcholine receptor at the neuromuscular junction of the nerve cells. Okay, like bacteria, how they can go and travel to your lungs, or the SARS-CoV-2, the COVID-19. Why is it so specific? It can only cause a breathing complication. I mentioned in the microbiology class, you are with me with the virus? No, you don't? You hate me, right? That's why you don't understand it. You don't want to learn more. So in that, okay, when you mention about the virus, their activity is very specific. We have a certain receptor that can recognize only specific type of virus. Like the SARS-CoV-2, they have this special ACE2 receptor at the lungs. That's why those people that got infected by the SARS-CoV-2 or COVID-19 virus, they will have breathing issues, sesak nafas. Okay, they don't travel to the brain. Another type of virus like this one, this are virus or rabies virus, they specifically go to the nerve cells. They don't go to the lungs. They don't go to the kidney. Because of what? Because they have a special unique receptors that they can recognize. Then they can bind. You know, in the matriculation center or CFS, you learn about enzyme and substrate, right? So they have to combine and bind together. Same goes here. The virus can only bind to specific receptors on our body. There are special protein that can only bind to the virus. All right. Same goes with some other virus too. Now you look back at my lecture. Last time I've already explained about this. Okay, now move on. I mean, not move on the slide. I mean, did we just discuss number 3? Why so early? It's not even half of the slide. Why you're rushing? Where you're going to? I know it's 10:19. You choose to have it at 9, but we don't have any other option. Yes, 4, 5, 6, 7. 7. Yes.
Okay. And the virus travel with an axon in peripheral nerve to CNS. And then at the spinal cord, the virus replicate in motor neurons and local dorsal root ganglia and rapid axon the brain. And finally, virus reach brain and cause fatal encephalitis. Encephalitis is an inflammation of the brain that can cause fever, headache, confusions, seizures, and in some cases, death. This phase can last between 2 and 10 days. The brain's then hippocampus is the important nerve cells that become infected, which cause coma and death. And finally, the virus spread to the skin of the head and neck, including the eyes, nose, and salivary glands. And saliva is infectious for up to 2 weeks before signs appear.
You want to wait for my question?
Okay, next slide.
Okay, now for sign and symptom. For initial symptom, Gopal might have headache, fever, fatigue, and tingling at the site of exposure.
Huh? What is it? Simple.
Symptom, typos.
Oh, symptoms. Okay. So this is the symptoms for the rabies.
Yes.
Okay. So you have the regular one like headache, fever, fatigue, and tingling. This is also common, like in the bacterial infection too. Fungus sometimes. But there are some specific symptoms, right? As I mentioned, what is the unique features that you can differentiate? Of course, you need to look at the sign and symptoms. And from here, you can see that this is very specific, especially the seizures. Okay. Most of the CNS related symptoms. Okay, so you can know about this. Like COVID-19. So what is the unique features of COVID-19? So you will know. So they must for each virus, okay, they must have a specific symptoms that you can relate on. Once you have this one, then you will go for the diagnostic point of view where you start to check for the blood or the presence of this virus in the body. Okay, move on. I don't see the CNS. So from here, where is the CNS? Only hallucinations? Yes. Delirium, what do you mean by that? Delirium. What do you mean by that?
Delirium is actually like a serious change in mental abilities. For example, like Gopal might have confused thinking or lack of awareness of his surrounding.
Okay, so remember, so CNS, most of them are related with the brain or spinal cord or the nervous system. Okay, the rest is maybe not so important, but I want you to relate with the CNS only at your level. Next.
Okay, now we will talk about pathophysiology of fever. When we were infected by a virus, the virus we also can call as exogenous pyrogens. When our body was infected by a virus, which is also called as exogenous pyrogen, it will induce our innate immune cell to release prostaglandin E2, pyrogenic cytokine, and also tumor necrosis factor. And these three will act simultaneously to induce fever. But the main one that cause fever is pyrogen. So pyrogen will flow from site via bloodstream, and the immune system identify potential trouble to the hypothalamus. So when hypothalamus detect the endogenous pyrogen, it will tell the body to generate and retain more heat so that our body will increase the set point of the temperature of our body and the fever will occur. Next. Okay, for the difference between bacterial infection and also viral infection. For bacterial infection, it's caused by bacteria, and some infection we can treat with antibiotic, but not bacteria are harmful to human. As we know, in this world we have good bacteria and also bad bacteria. And for example, for bacterial infection, it's just like TB, urinary tract infections, and also impetigo. And for viral infection, it caused by viruses, and it can be treated with antivirus, but can't be treated by antibiotics. And viruses cannot reproduce outside of living cell because for virus to reproduce, it needs a host cell. An example for viral infection is like COVID-19, HPV, and chickenpox. And for both infection, both can cause harm to humans, and both also have similar transmission route and similar prevention measure. And we can take vaccination to prevent the infection. Next.
Okay, classification of rabies virus. Rabies virus belongs to the order mononegavirus virus with a non-segmented negative stranded RNA genomes. Within this group, virus with a distinct bullet shape are classified in the Rhabdoviridae family, which includes at least 3 genera of animal virus, which is Lissa virus, Epimerovirus, and vesiculovirus. The genus Lissa virus includes rabies virus, Lagos bite, Mokola virus, Duvenage virus, European bed virus 1 and 2, and Australian bed virus. Okay, the structure of Lissa virus, Rhabdovirus are approximately 118 nanometer long and 75 nanometer wide. The rabies genome encode 5 protein, which is nuclear protein, phosphoprotein, matrix protein, glycoprotein, and polymerase. All Rhabdovirus have two major structural components, a helical ribonucleoprotein core (RNP) and surrounding envelope. In the RNP, genomic RNA is tightly encased by the nuclear protein. Two other viral proteins, the phosphoprotein and the large protein, L protein or polymerase, are associated with the RNP. And next slide.
So what type of the virus?
What type of virus? Rabies virus.
That's the name of the virus.
Alisa virus.
No, the type. We're going to have another 2 hours virus lecture. We have two type of virus, DNA or
RNA.
RNA. So this one is
Single strand RNA.
very good. Okay, single-stranded RNA virus. So what's so special about RNA virus as compared to DNA?
The negative positive.
What happened with the positive negative? It's not that important, negative and positive. The most important, you know that the RNA, okay, you need to know the central dogma. Have I taught you about central dogma? Inside the nucleus of the cells, like human cells, we have the nucleus, right? Inside the nucleus, we have what? The chromosome. 23 pair of chromosome, total 46. Okay, the X, like a letter X, okay, that is paired, X and X, right? So the chromosome is what? Combination of genetic information in the form of DNA. So the function of the DNA is to store the genetic information to produce the protein. Whatever protein, maybe protein for new cells, maybe protein for new hormones, maybe protein for new molecules in the body, whatever protein. So every single protein that has been produced in your body, okay, is encoded by the DNA. All right, so DNA is stored information for your body to produce new cells so that you can get bigger and bigger. Okay, so since you are a baby child, then you grow, right? So the information from the DNA, it keeps on producing protein. So the function of DNA is to produce the protein, essential for your growth, for your physiological function, for structural anatomy. But what I want to highlight is that the central dogma is the fundamental principle that says that DNA must be converted into the RNA, which means that DNA is in the double-stranded form. They need to be converted into RNA, which is single-stranded. So the DNA will be transported into RNA or be converted into RNA, which is going out from the nucleus. So the DNA is inside the nucleus. Once it's become single-stranded RNA, they will travel into the cytoplasm. Okay, so why these things happen? This is the central dogma. It means for proteins to become or to be synthesized, they need to be converted from chromosome in the form of DNA in the nucleus. They need to become transcribed into RNA out from the nucleus. Now their RNA is in the cytoplasm, and RNA will travel into the ribosomes, you know, the organelle in your cells, the ribosome. So inside the ribosome, this single-stranded RNA will get into the ribosome and the translation of new protein will occur. If you look at the Google, you type central dogma, D-O-G-M-A, you will find that this is the process. Okay, DNA will be converted into RNA, and RNA will be converted into the protein. There is no way that the DNA can be converted directly to protein. It must have this process, okay? DNA, RNA, and also protein. In the case of virus, we have two types of virus, DNA and RNA. If it's RNA virus, it means it bypassed the conversion of DNA to RNA. So now it's in the single-stranded, it's easier to get infected by you. Okay, so it doesn't need an extra step, which is the first step, DNA to RNA. So RNA virus like COVID-19, SARS-CoV-2, right? Coronavirus, they're all RNA virus. That is very dangerous because the effect is quicker. Because the process from DNA to RNA is not there anymore, because it's already in the form of RNA. Once you got the RNA virus infected, immediately this virus can turn into a protein structure that is harmful to your body. All right, so this is only one step. But if it's DNA virus, it takes longer time for your body to get the symptoms or the effect. Understand the central dogma? Yes? Okay, so next time when someone say what's so important between DNA or RNA virus, you can say about the time that they can infect people. Okay, it's more quicker because it's only one step. If it's DNA virus, it has to go two steps, DNA to RNA first and then RNA to the protein. I hope that can clear the confusion. Okay, next. How many slides left? Because my bed is calling now. This is the last one. Why not? I told you to only produce how many slides last time? Only 10 slides, right? Did I told you that? Next time, make sure you just comprise or cram them into only 10 slides. Only 10 slides for each presentation, PBL 1 and PBL 2. Sorry, not PBL 1. For each PBL, scenario 1 or scenario 2, or trigger 1, trigger 2, each of them should be 10 slides max. So what we're having here, lab diagnosis. So this is basically the way that we can detect, yeah, in the lab. Okay,
Yes.
tell me, yes.
We have a saliva test. A sample of your saliva is tested for the presence of the rabies virus. Saliva can be tested by virus isolation or reverse transcription followed by polymerase chain reaction. Then lumbar puncture, a needle is used to remove a small sample of cerebrospinal fluid, CFS, which can be checked for the rabies antibodies. Serum and spinal fluid are tested for antibodies to rabies virus. Then for blood tests, blood is checked for the rabies antibodies, which is rabies antibodies test is a sample blood test performed on a sample of blood to evaluate the level of rabies antibodies in the blood. And then the last one, examination of brain tissue by using either PCR, fluorescent antibody to rabies virus. Here we use the tissue, and then we use a microscopy examination to make a treatment, not a treatment, but we use a nervous tissue after treatment with anti-rabies serum or globulin conjugate with fluorescent isothiocyanate.
What is
That's
very
all.
complicated, our scientist Alia?
Huh?
Alia,
Yeah.
Hafiza. Okay, which one is, we start in the hospital, which one comes first? Are we taking this all at the same time? Which one is, the first line? We start with what?
Saliva test
Saliva, we call
or
it.
blood test. Saliva.
Could be saliva, because saliva is not that accurate as compared to the blood, right? So maybe the doctor may ask you to, ask the patient to take both at the same time. This one is just like the RTK kit you have for the COVID-19, but it's not that accurate because it's a wide range antigenic detection. It can be, okay, it can also detect some of the coronavirus too. So it can cause like false positive results. So to further confirm that this is really rabies virus, you take the second test, which is the blood test. Okay, in the blood test, you will check the level of antibodies. So when you mention about the antibodies here, what type of Ig or immunoglobulin that is detected for this virus? We have how many antibodies? $63. How many antibodies? 63. Have you learned about this immunology before? Don't say no, because you already got vaccinated with all this kind of stuff previously. You have your own immune system. You must know, you get it, the milk from your mom since you're a baby. When you come to the planet Earth, they're full of antibodies. So my question is, what type of antibodies for detecting or confirming that it's a rabies infection? Okay, it's okay, you cannot answer now, but you tell me in the group. Okay, this is the first question. The second one, lumbar puncture. When you mention about a needle, is used to remove a small sample of the cerebrospinal fluid, which can be checked for the rabies antibodies. So this is quite a later stage, right, Nabila? Sleepy already. So you isolate this CSF to check why we take this CSF. Okay, that's the second question. All right, the first question, what is the Ig or immunoglobulin? How many immunoglobulin or antibodies that we have, and what is the specific antibodies to detect or to confirm the rabies virus? The second one, why we take out the CSF from Gopal. And then finally, one is the PCR. PCR, and this is also true for the coronavirus. Tell me simply, what is the PCR? In the simplest explanation, I want it to be your own words, so based on your understanding. Okay, keep that 3 question. Answer it in the group, WhatsApp group for that one. Okay. Each person have to answer them. Okay, 1, 2, 3. You put your answer in the WhatsApp so that I can know. Use your own words, how you translate based on your understanding. Don't copy and pasting. Next. Okay, you must be, expert in this.
Yes. Okay, Assalamualaikum warahmatullahi wabarakatuh. Sekarang, now I would like to continue the treatment and management after we get bitten by the dog.
Okay, so for the first treatment of rabies, after we get bitten by the dog, don't go lay back and watch TV, but instead we go wash the bites or the scratch using the soapy water, iodine or detergent for like about 15 minutes. Why? Because this might minimize the number of the viral particles, alright? But then after then, you cannot like wait for the symptom to come to you or like to show up, no. But you need to seek for the medical help. Go to the clinic, the nearest clinic, go seek doctor, right? So after exposure and before the symptoms begin. Maksudnya pakai gigit terus pergi jumpa doktor lah. Okay, so a series of shots can prevent the virus from thriving. Okay, shot apa? A fast acting shot, it called as rabies immune globulin. Ataupun HRIG to prevent the virus from infecting. This is given if you haven't had the rabies vaccine. Sebab kalau like if we had the vaccine, our antibody will fight the rabies, right? Can. So this injection is given near the area where the animal bit you if possible, as soon as possible after the bite. Yes. So the second is a series of rabies vaccine. So this vaccine will be injected into the arm for over the next 2 to 4 weeks. So apa yang vaksin ni akan buat? It will train the body to fight the virus whenever it find it. So it is safest to assume the worst. Kita kena rasa macam all animals have rabies. Sebab tak semua animal kita suntik kan, right? So kita kena assume the worst. Yes, that's it.
Yes, but what is the drug? You mentioned about the treatment. Where is the drug? What type of drug? Mention about the treatment. What type of the drug? Come on, everyone. Chicken, where's the drug? Just put your eyes on the screen and you can see the answer.
Human rabies immunoglobulin.
Brilliant, Dr. Yes. So that is immunoglobulin I mentioned just now, but we don't know. Okay, this is what? Immunoglobulin is what? AKA? Immunoglobulin AKA? AKA?
IG.
Yes, IG, AKA?
Antibodies.
Very good. Okay, that's right. Antibodies. Okay, not antibiotics. Okay, it's antibodies. Antibodies. So immunoglobulin is an antibodies. But this is virus. Is it sufficient to cure this by taking the immunoglobulin? I don't see any antiviral drugs. What do you say on that? For bacteria, you prescribe antibiotics, but why you didn't put antivirus or antiviral drug here? Is it still not? It's not ready, or what happened? Because the rabies infection is quite common. Is there any drugs for rabies? Any antiviral? Apart from the antibodies? No? You don't find any? You find that antiviral drugs for rabies? You tell me. That's the fourth question. Okay, next. The other one you mentioned about the vaccine. Okay, acceptable.
Okay, after the first treatment, you will have the prevention of the long treatment. It's say like to prevent the wound getting worse. So first,
you know that the dog bite can be introduced into the dangerous virus and it may cause fatal infection if you leave it untreated. As the first treatment, you will wash the wound, and then the one you must to change the bandage of the wound daily and keep the one from any dirty surface and keep your eye on the one if there is any sign of the infection, because the infection of the viral infection can be spread quickly, and if you notice the infection getting worse, you will have to contact with any doctor and go to emergency so the doctor can provide you some antibiotic to make your wound didn't getting any worse. There is some rabies prevention. As you can see here, you can make sure that your pets getting vaccination is up to date. If and you can leave alone the white animal because we didn't know that they have any vaccination or not. If you are at the high risk of the rabies, you can go to see the doctor and get some tests of your infection. Okay, next.
Okay, hemostasis is a mechanism of response that stop bleeding. It consists of three of them: vascular spasm, platelet plaque formation, and blood clotting or coagulation. Okay, first the vascular spasm. When we get like injury, like our blood vessel are damaged, there's like the circularly arranged smooth muscle in the vessel wall. It will contract immediately near the injury point. This is to reduce the amount of blood loss. For platelet plaque formation, there's 3 steps: platelet adhesion, platelet release reaction, and platelet aggregation. For platelet adhesion, the platelet contact and stick to part of a damaged blood vessel. For example, collagen fibers or connective underlying the damaged epithelial cell. For platelet release reaction, the platelet will become activated due to adhesion and their characteristic will change very dramatically. They extend many projections which enable them to contact and interact with each other. Then they begin to liberate the content of their vesicles. Also, adenosine bisphosphate, ADP, thromboxane, and serotonin will be released to activate other platelets and sustain vessel contraction and decrease blood flow. For platelet aggregation, other platelet in the area will become sticky due to the release of ADP, and the stickiness of newly recruited and activated platelets causes them to adhere to the originally activated platelets. And eventually the accumulation and attachment of large number of platelets form a mass called platelet plaque. Okay, next.
Why are you showing this slide to me?
Because on previous PBL trigger 1, you asked for us to talk about this.
Yes, why we talk about this? Why you show me this?
Because like Gopal got bitten, so like makes sense to talk about the hemostasis because you don't need to.
Fantastic. Okay, so you know that this is an injury point of view.
Yes.
So that is more to the injury and also the blood coagulation, the formation of a closure or the closing of this wound site. So that's why it's relevant to be put forward. And we need also to understand the process And basically, this is the step-by-step process. But I want you to tell me, okay, what is the thromboxane or this ADP, serotonin? What are they? What is their role in this activity or in this process?
To activate other platelets and sustain vessel contraction and decrease blood flow.
Are they considered under what molecules? Are they blood coagulators, or are they signaling proteins, or are they hormones? You tell me, what are they? In B, in 2B, platelet release reaction. Why do we need to release all these ADP, thromboxane, and serotonin?
Apparently, it is a neurotransmitter, but also act as a hormone.
Serotonin.
Mm-hmm.
Serotonin is just like acetylcholine. You mentioned about neurotransmitter. And at the same time also work as hormones. So what did they do? Serotonin. Kiha, serotonin. You have serotonin or not? You think you have? What does it function? What is the role of serotonin?
Carrying message from our brain to our body.
Again, what do you mean?
Carrying message or what we call impulse throughout our body, from brain to throughout our body.
Serotonin. Camelia, do you agree with Fakiha? Camelia?
Yes.
Agree? Why do you agree with her?
Because serotonin play a key role in body function as mood, sleep, wound healing.
Okay, so that is on for the mood, sleep. So if you going to bed after this, is it high or low?
Low.
Are they going to become low? And then you can sleep. Yes? Okay, that is for sleeping. But we are talking about the hemostasis or the closure of the wound site. What is the function of serotonin in this case? What is the function of serotonin in making sure that the blood or the wound is properly closed?
To minimize the blood loss.
A bit general. Was there any specific action of serotonin?
Promote cellular viability.
Again. Promote what?
Promote cellular viability.
Camelia, can you shout? Can you shout? I cannot hear you.
What? Cellular viability.
Cellular what?
Viability.
Dia what? Can you write it down? I can hear someone is talking, so cannot hear your voice clearly. To promote what? Cellular viability. Viability means life of the cell, or as they will die. Okay, so you mentioned about the serotonin. So that's also a bit general. in the case of the 2B platelet release reaction? Still doesn't answer the question.
Doctor,
Yes.
serotonin to induce constriction of injured blood vessel and enhance platelet aggregation to minimize blood loss.
Fantastic, as she came. So now it relates with the blood vessel itself. Okay, two only mechanism that you can relate with the blood vessel, whether it's vasodilate or vasoconstrict. So the function of serotonin is to what? To what? Constrict or to dilate more? Nasratul. Serotonin is released to do what?
To constrict the injured blood vessel.
Brilliant. So when you constrict the blood vessel, what happens is that more or less blood coming out?
Less
blood. Okay. So you will put a pressure there. That's why if you got an injury somewhere, okay, it's good to seal it. Okay, you need to put some bandage, prevent the blood from coming out from your body. Okay, so you put a stop there. So, or you put a pressure. If you don't have any bandage, or you just put pressure there, this is to stop the blood from coming out. Okay, but serotonin functions to do this on their own. All right, it's gut release, and then it will act on the blood vessel by constricting the endothelial cells of the blood vessel. Now the diameter is become narrower, so lesser blood will come out, and it also can facilitate earlier or quicker healing of that site or injury site. And of course, here there are a few processes because you need to close it. It's not, when you get injury and then you immediately got healed. Of course, you can see that after 1 hour, you can see if it's just a minor cut, when you potong bawang, for example, then you just cut your finger. All right, but this is very minor cut. It can heal just within 1 or 2 hours. All right. But in the case of Gopal, maybe the bite site is a bit severe. So it's always need to take good care and check for the severity, what is the damaged cells, whether it's deep down to the muscle layer or is it only on the dermis or epidermis layer. It can dictate or it can decide determine the severity of the disease. Okay, so if the dog bites on the bone, for example, deep down into the bone, and it's more severe, right? If you compare if it's only on the muscle side or muscle tissue, because the deeper it goes, okay, the more time you will need to recover. And of course, the injury or the blood will come out more. At the same time, the rabies virus will spread in a greater scale. All right. And finally, you have these platelets to come and seal it at the first layer. And after that, lots of other proteins. We call it the mesh protein or fibrin network will come and seal it like a mesh there. So it will stop the bleeding. In the worst case scenario, where if the wound is very big and very severe, there might be some collagen rearrangement. Maybe you get like some keloids or macam parut tu. So that is a more severe condition. All right. Move on.
Okay, for a blood coagulation, there's a 12 factors of blood clotting. For the first factor is fibrinogen, to adhesive protein that forms the fibrin clot. Second is prothrombin. So activated form is main enzyme of coagulation. The third one is tissue thromboplastin, to lipoprotein initiator of intrinsic pathway. For the lipoproteins, it carry cholesterol and triglyceride to cell in the body. The fourth is calcium ion. The function for calcium ion is metal cation necessary for coagulation reaction. Next, proacerine, to cofactor for activation for prothrombin to thrombin. Next is proconvertin with tissue thromboplastin initiate intrinsic pathway. Next. Okay. For the hemophilic factor, the function for this factor is cofactor for intrinsic activation of factor X. Next, Christmas factor or anti-hemophilic factor B. To activated form is enzyme for intrinsic activation of factor X. Next is Thorpe-Prower factor. So activated form is enzyme for final common pathway activation of prothrombin. Next is plasma thromboplastin antecedent. So activated form is intrinsic activator of factor for the factor IX.
What is IX?
Christmas factor or anti-hemophilic factor. Okay, next is a Hageman factor that normally start a PTT-based intrinsic pathway. PTT means partial thromboplastin time. And the last one is fibrin stabilizing factor, transaminase that cross-links fibrin clot. Next.
Why you skip the Quran? You don't like it? What do you want to say here?
Alright, from the I think it's from the hadith, another iman, which we can say is cleanliness is a part of our iman. So by we take care of our cleanliness, we can avoid further infections, or worsen the infection that we have. There can be extra infection like bacterial infection, which is like maybe you can have trough, sore, or scratchy, or it can lead to kidney inflammation or rheumatic fever.
Fatima,
Yeah.
this is Gopal, right? Gopal being chased by the dog, and then you ask him to observe the cleanliness. How does it relate?
Cleanliness is when he have been bitten by the dog. So when after he get the treatment, he have to take care of his cleanliness so that there will not be any further infection occur.
No, this is not everyone know because Gopal is not Muslim, right? So he know about the cleanliness. Every single person, whether it's Muslim or non-Muslim, they know that they have to become clean or, make things clean. But what is the Islamic perspective when you're being chased by the dog?
We remember Allah.
Yeah, something like that. Try to relate with the scenario, not necessarily with. I don't want you to take Quran or Hadith to bring forward because it's a bit deviating from the discussion. The Islamic perspective is not about the Quran or Hadith or what the scholar says. It's about how do you think that the God is observing you. You think that, what is the Islamic value that you can integrate in the lesson or in the case. Because if you put like in the Zofatu Minal Iman means, doesn't mean anything for Gopal because he don't read Quran, right? So I want you to, understand what is the Islamic value that you can extract when you're being chased. Just imagine you are part of the, of the scenario. So maybe, yes, you can say something, right? What is it?
Of course, we will if we are in a bad situation, like getting chased by a dog, we will remember Allah, because that's how human is. If we are in that situation, we need helps, we will try we will like try asking for help. So we will remember Allah and we say, oh Allah, please save me from this dog, and that's it, running away, so on, and ask from Allah prevention from getting beaten, if we have getting beaten, from getting infections that can maybe lead to death, maybe if the infection worsens. So what more?
Yes. So this is one of the things. That's why I want you to give a reflection. It's not about putting some words from the Quran. No. Okay, I want you to say something like this. Okay, maybe you can say, you can start to remember Allah, ask, dua, and then you tell me. Then the dua is like this: Allahumma ajurni fi musibati wa akhlif li khairan minha. Okay, and then you give out like the doa, the suitable doa related to that situation. But the most important is how you reflect on the situation, not taking pabrikjammasjid.com something. This is Indonesian.
Alright.
Okay. All right, move on. What is this?
No, this is the last time slide. So
Okay, you don't need this one, right? Okay, so we are done. Thank you. It was very good discussion for tonight. Suppose that I have put down some of your marks actually for the your performance during the presentation, and also your reflection. How do you want to interrupt? But I couldn't see. Only few people managed to interrupt, or to help your friend when I start asking the question. But please keep it on, because next time I will start to evaluating you again and again till we finish the PBL. Remember, your personal or your individual performance is very important for your carry marks too, apart from the group work. Okay, so that is the end of the fight or flight response for Gopal. I think I can explain you further, but after this, maybe you need to compile all these things, all this literature and all this information into the Word format. So if you can write a report and send it out to me through email, it will be great. I just need a Microsoft Word. Okay, so you put the name of your group members in the front, put the metric numbers, and you put the scenario, or you put your lecturer's name. And for the second page, you start to list down all the things that we have discussed. Like from the first scenario, you put your fila, and then after that, you put some introduction. You discuss about the virus, okay, the mechanism, pathophysiology or pathogenesis, how it travels to these sites at the neuromuscular junction. And after that, you go to the maybe some of the anatomy and physiology. You mention about the hemostasis or the blood clotting mechanism. And after that, you proceed with the treatment and management of this patient. Okay, so you put all the signs and symptoms, all the related medical advices. You compile it together, distribute the job within yourself. Okay, you just put in the Microsoft Word and then send it out in the Microsoft Word format. Okay, apart from that, you also need to send an email that compiles all the presentation too. Okay, so I will give you time before we have our next PBL presentation, which is PBL 2 for trigger 1. I think it's gonna be next week, if I'm not mistaken. Are you allowed to have the online version of class next week? Are you coming to the kuliah or not? Why not?
No.
What happened with you? We have finished with the voting, right? So why don't you come back early to the kuliah? What happened?
Continue with the mid-sem break. So
When is the mid-sem break?
26 until 4.
Until 4th of December. Are you allowed to have class during this mid-sem break? Are you not allowed to have class? Or you just want the break? Can we have the PBL during the break? Yes, okay. All right, so for the next PBL, I just put it into the checkbox function. And then we have this Mark Wynn, he, not she, a 42-year-old travel vlogger, recently involved in the volunteering works in Animal Conservation Center in India. He has a history of type 1 diabetes mellitus. This is the genetic predisposition. Presented to the IUM emergency department, the ED department, with fever 38.5 with retroorbital headache and nasal stuffiness. Is there any information that you require from this first trigger for PBL2? I believe that you need to sit down again within yourself, try to look and fill in the FILA table, what would be the best facts, ideas, learning outcomes, and also the action plan for this patient. I think you know Mark Wynn, right? You don't know him? Vlogger? I didn't know him too. Maybe this is just a fake account or fake person. Okay, so any more information that you require from this trigger? You can ask now.
Is he a fat person or thin person?
He's about my height and my weight. My height is 170, my weight is 69 kg. Actually, try to be honest, but actually it's 75 actually. Okay. Anything else you want to ask? No? Okay, so he has a type 1 diabetes mellitus. I hope you know about this. This is the genetic type of diabetes mellitus that occurs during childhood. It's not like the type 2 or, the pregnancy induced or gestational diabetes. And he been admitted to the ED at IUM with fever, retroorbital headache. So you tell me, what is retroorbital headache and nasal stuffiness? What would be the possible diagnosis? Your job is not to diagnose, actually. Your job is try to narrow down the discussion and find the what we call the factors that cause these symptoms. I would like you to look at this pathophysiology related with this patient. What are the risk factors? What are the possible diagnoses? Some complication maybe. All right. So this is still superficial and it's still not enough for you to know the exact diagnosis for the patient. But as I mentioned earlier, if you have an infection, you can tell whether it's fungal, bacteria, or virus. If there's no more question, I think we will stop up to this point. You will discuss this with me. Try to remind me again when is the best time to have this first round of presentation for the PBL2. We will do it online throughout your mid-semester break. So are we going to have any class next week? I mean tomorrow, Monday, right? You still have class, not regular one. Or is it already mid-semester break? When again? When is your mid-semester break?
26.
26, yeah. Oh, so we have another one. Okay, so next week, between 21st to 25th of November. So perhaps we can have one class for the hemoglobin and hemoglobin synthesis for biochemistry. So if you are in the section 1, 2, or 3, please, if you can tell your class leader to contact me so that we can have an online session for that class. Maybe we just combine like usual, all at once. Okay, section 1, 2, and 3 at the same time. Please, if you can help me to tell your class leader for that to contact me tomorrow and onwards. Okay. Any more question before we end the session? Good, good, good. I think all of you passes for this second trigger. I'm quite happy with your performance today. But please, if you can increase your sense of belonging, try to help your friend if you see him or see her struggling to answer the question. Okay, you can just unmute, say, I don't think like that. Okay, you can just give your opinion. It's not right and wrong in my class. It's just how you defend your arguments. Till we meet again. Bye-bye, everyone. Good night. Assalamualaikum. And we end the session by Tasbih Kafara and Suratul Asr. Subhanakallahumma wa bihamdika asyhadu an la ilaha illa anta astaghfiruka wa atubu. Wal 'asr inna insana lafi khusr.